Okay, so this week we're going to be discussing a number of techniques that are used to study the brain, and understanding the techniques, both their strengths but also their limitations, is very important in evaluating, research that's based on, the utilization of these methods. And we're going to be looking at neuroanatomical techniques, which are going to be, examining the actual structure of the brain, which depended, on the development of microscopes so we could see aspects of fine neuroanatomy, staining techniques so we could actually visualize individual cells, for example, tracing techniques so we know what is connected to what, and just sort of a parsing of, how the structure, of the nervous system actually relates to its function. So neuroanatomical techniques are going to be very important. We're going to also talk about electrophysiological techniques, which are based upon the fact that neurons carry, messages in the form of action potentials, in the form of electricity, of the movement of currents, right? And so, this is going to involve, multiple cell recordings, direct electrical stimulation, sort of aggregate recordings like EEG, and ERP. So we'll be talking a lot about electrophysiological techniques, and we'll be talking about imaging techniques too. And some people might say that an EEG is an image, actually, of functional image of what's going on in a brain. But we'll be talking about for example, MRI and DTI. Some of these techniques that can measure structural changes. DTI, we'll learn, for example, is important for tracing white matter tracks in the brain. But also techniques like resting state functional connectivity, MRI, where we actually can see how distributed networks in the brain are kind of functionally organized and are, changing in concert with each other. That indicates, sort of collective, kind of coordination. And then we're going to be talking about genetic techniques, including, for example, CRISPR, Cas9. There were some Nobel Prize winners in 2020, for the development of this technique, and a lot of other genetic techniques that have gotten more and more powerful in terms of our ability to sort of manipulate, genetic gene expression and protein synthesis. So this week is going to be all about techniques.